Hey guys, so thanks for having me. So this is Singh. I'm in electrical and biomedical engineering, and a lot of my work is primarily mathematical modeling. So I did work in a wet lab before, but now I run a purely dry lab where I collaborate a lot with people. So this is a great platform to talk to you guys a bit about my research and explore collaborations. So today I want to talk about some work we're doing in single cell biology. And as you know, even within the same cell population that's clonal, you begin to see heterogeneity in expression and phenotype between cells. And that's what we're trying to quantify both experimentally and using mathematical models. And it turns out that this variability between cells is biologically important. And by studying it, we can learn mechanisms that are not possible in population average data. So that's a kind of a big theme of kind of our research. So I want to show you some examples of our kind of collaborations, looking at this in cancer cells and in immune responses and in virus infections and so on and so forth. Okay, so I generally give my students this kind of video, which probably you've seen before, right? So basically, in our lab, we want to understand how single cells use complex biological networks to sense information, process it, and make decisions that leads to complex phenomena like a white blood cell using chemotaxis to capture the bacterial cell, right? And then we collaborate with different life scientists doing experiments, and we mostly do the computation, imaging, mathematical modeling, and those things. So basically, here's kind of a very simple view of how cells make decisions, right? You have complex networks of signaling, gene regulation, and a cell will process environmental cues and make a decision. And at a single cell resolution, it's really amazing how remarkable two cells can be, where two cells from the same mother can make different decisions to the same stimuli. That's the noise or randomness we're trying to quantify, understand where it comes from, what are its consequences for biology and medicine, and what can we learn from it by combining it with mathematical models. So an important source of cell-to-cell variability within the same population is the fact that expression of any gene is somewhat stochastic, right? So the level of a given protein in a given cell can fluctuate over time. So red is a single cell expression, blue is the population average. So each single cell can deviate from the population average in its expression pattern and hence its phenotype. And these fluctuations come from the fact that you have very low copy numbers. So in a human cell, you only have two promoters of a gene, for example. In a microbial cell, an expression of a gene can be a few mRNA molecules per cell. So at that low copy number, you begin to see this enhanced fluctuations in expression levels that create phenotypic heterogeneity within the same population. So I'll show you some actual data on expression from bacterial cells and mammalian cells. So here's the most kind of seminal paper in the field about 20 years ago, right? Where they had this very elegant experiment where you have two identical promoters driving different reporters. So let's say you have GFP and RFP, and they put both promoters inside the same cell, and they see this remarkable heterogeneity at a population level where each cell has both promoters, they're identical, and they make both proteins, but some cells are red, some are green, some are orange, and some are yellow. And the fact is that because expression is somewhat stochastic, you have fluctuations in protein levels, and depending on the time point and the ratio, you can have cells that can appear in different colors. So this paper has been now cited at least 8,000 times, last time I checked. and published in the early 2000s. And since then, people have kind of shown this kind of noise and expression in pretty much every cell type, right? From cell lines to primary cells to plant cells. And a lot of my research is trying to mathematically model it and understand its consequences in the context of how does a cancer cell evade chemotherapy, for example. So I'm going to show you some examples of that in a bit. Here's some data on yeast. So you have two alleles, right, of the same gene. A green dot is a single mRNA molecule of one allele, and the pink dot is the mRNA molecule of the second allele. So you have two exact alleles, same alleles in the same nucleus. They're expressing, and you have fluctuations in mRNA levels. So on average, you have 5 copies per allele. That's the low copy number I mentioned. And you have fluctuations in mRNA levels that are not correlated. That is, it means it could be random timing of transcription at the two different genes. And then each transcript will make 100 proteins. So these fluctuations will then propagate upstream to the protein level. And finally, here's some of our own work in PNAS with Sanjay Tyagi looking at expression in HeLa cells. So here is a HeLa cell, and we're doing RNA fish on a specific gene of interest. So in this cell, 2 copies of that gene are on, and you can see the red is the mRNA buildup in the nucleus and cytoplasm, and the neighboring cell only has 1 copy on, 1 copy is off, and this cell has both copies off, but you see mRNA, which means that they must have been on in the past. So at this time point, you see this heterogeneity where different genes are active, they're turning on, they make mRNA, they turn off. And in this paper, we showed that this opening and closing of chromatin, right? It plays a crucial role in this kind of bursting behavior where each allele turns on, it makes a bunch of mRNAs, it turns off, and it does it stochastically. And that's why you see this heterogeneity where the number of copies on varies between cells. So, okay, so expression is stochastic. It creates this heterogeneity, but is this biologically important? So I'm going to show you some examples from our research where we try to connect this to, for example, cancer treatment. So here's some work that we do with Arjun at UPenn. We look at drug tolerance in melanoma cells. So this is basically a population of melanoma cells. So each dot here is a single cell. The cells are mostly blue, so these are drug sensitive cells. And cells that are colored, for example, in red and yellow, have high expression of a gene that makes them drug tolerant. So even when there is no drug in the environment, a small rare subpopulation of cells is primed to be drug tolerant. And these cells, they arise transiently and they can revert back. So this AXL positive cell over some cell cycles can become AXL negative, and an AXL negative cell over time can become AXL positive. So it's like a reversible switching. So what we show in this paper is that cells are mostly drug sensitive. They can become drug tolerant reversibly and switch back. And that will prime the subpopulation to be tolerant. And when you hit them with some drug therapy, these cells survive. And as they survive, they can pick up mutations that can lead to a stable resistance. So basically, upon therapy, you see the tumor size going down, it will stabilize and then relapse. And these small primed cells play a critical role in this drug resistance. Interestingly, we also saw examples where you have 2 sisters from the same mother before you add the drug. After you add the drug, they have different phenotypes. So this cell stops proliferating for about 3 weeks. This sister keeps proliferating and forms a resistant colony. So I think it shows you how much the role of noise and expression is. So for example, this sister could have a burst of a drug tolerant gene, and that made it tolerant, and then it evolved resistance, while the other sister, which is identical in every other way, was not able to proliferate. So here are some papers from our collaborations, mostly in melanoma and breast cancer. We are looking at this stochastic variation and its implication for drug tolerance. And here's kind of our modeling approach to understand how these cells arise. So as I mentioned, each gene can turn on and off. It can make mRNA, and these genes are interconnected in some circuit. So now think of this gene as a drug-tolerant gene, and you have some topology of connection. And we show in the paper is that with the right topology, you can get situations where in single cells, different tolerant genes get expressed at the same time. And these are the drug-tolerant cells. And then over time, this can be lost, and they revert back to being drug sensitive. So basically, some kind of mechanistic modeling of gene networks can help us understand how these rare cells arise and arise transiently and then go back to become drug sensitive. Here's some recent work, same idea, but in bacterial cells. So the first paper is coming out in I think, Spectrum, where basically we show that E. coli cells are also primed for survival even before you hit them with some lethal antibiotics. So when you add antibiotics, say 1 in 1,000 cells survives, and those cells are non-genetically primed for survival. And we also showed in a similar paper that cells have memory for iron, and this memory for iron has different consequences in terms of antibiotic responses, biofilm formation, and swarming. So two kind of papers looking at the same idea of stochastic variation in bacterial cells that could be interest to you guys. And then this is a paper where we work with Sergi Rigaud at Johns Hopkins looking at immune responses. So again, you have human epithelial cells, and again, they're all clonal, they're all infected. But what we see is that the immune response is very calibrated, where only a small fraction of cells upregulate the innate immune response as measured by NF-kappa B activation. So only say 15% of cells are responding, even though all cells are identical, they're in the same environment, and they're all getting stimulated. So what we show in the paper is that there is this epigenetic priming that happens in these cells that ensures a response which is calibrated, because you can't overshoot an immune response and you can't undershoot. So this epigenetic priming plays a crucial role to calibrate the right immune response to the pathogen. And then we had a follow-up eLife paper where we actually found that this fraction that respond is somewhat density dependent. So we think there's some kind of quorum sensing in the immune response where the fraction of cells that upregulate, in this case interferons, depends on the density of cells that are around. And then finally, I want to talk about a bit about some of work in phages. So we love phages because it's a nice organism for doing modeling. And we work with all kinds of phages in different types of bacteria. This is the classical like lambda phage, right? Where you infect E. coli and you can have lysis or lysogeny. And then we have a project where we reactivate lysogens and look at the time of reactivation in single cells. And this was my first R01 grant almost 8-9 years ago to look at timing in cells at a single cell level. So you take a lysogen, you do thermal stress, you reactivate, you look at these cells in a microscope, and you find the time it takes to lyse. This is what a movie looks like, right? You have a single cell which has the phage reactivated. I'm showing the last 5 seconds of this movie. So here at this point over here, you see this kind of burst, a collision here, which is a hole in the membrane of the cell, and the cell would then lyse. So then we can watch these movies for different growth conditions, different genetic strains, and get the timing of lysis. And here's the actual data on the timing of lysis in these growth conditions. It takes about 1 hour with some standard deviation. And we think it's quite precise. The timing is quite precise because within 1 hour, plus minus 5 minutes, almost all the cells will lyse. And then we are building a mathematical model based on this protein called holin. So holin is a lambda protein that will accumulate in the cell membrane. And when you have some critical threshold, it will come together and form a hole. And when the hole forms, is when you get this kind of burst of the membrane and the cell would lyse. So here are some kind of papers from our group modeling different aspects of the timing of lysis, hole-in-hole formation. We're looking at a number of phages made per cell and how stochastic that is in the same population. We're looking at more mathematical methods of modeling timing phenomena in cells. So here's kind of a group of papers that you may want to look at if you're interested in that question. And finally, here's a paper that we are working with a collaborator on embryonic stem cells, where again, in the same population, these cells can fluctuate between different states, and these states prime them to differentiate in different pathways. And then we have a network of transcription factors that regulates these fluctuations in states, and then you can perturb those networks and change the frequency, right, of different states. So again, a very interesting project with somebody called Salil Garg at Yale. We're trying to understand how these states arise, what's the time scale of switching, and how that time scale is controlled by the underlying gene network of the cells. Okay, I think that's a kind of brief overview of the different projects. So again, as I said, I collaborate a lot with different people. Here are a list of some people. So work with lambda phage is done with John Denny in New York. The work with cancer cells is done with Arjun and Sydney at UPenn, and a lot of different collaborators here. And I would love to have some of you guys to collaborate on these projects. Some of the lab members we have currently, we have 3 postdocs and 5 students. And I also want to acknowledge some of the funding that we have from mostly NIH and NSF. So, okay, I'm going to stop here and maybe take some questions.
Great.